Hello and welcome back to Microbial Concepts. So here is another video related to immunology and that is on antigen. Okay, so today we will be only focusing on antigen. Okay, so in comments I got this suggestion for making a separate video only on antigen. So here it is. So let's start. So first we will see what is antigen. So you learn a lot about antigen antibody reaction, correct? But here we will be just focusing on antigen. So why these are important or why we learn about antigen? Okay, so we will see that. So antigen is a substance that can induce an immune response. Okay, so once antigen enters in host body, it will start inducing some reactions, okay, some immune response. Okay, so the substance which does this, which induces or which triggers immune response is known as antigen. So substances that can be recognized by the surface antibody, that is on B cells, or by the TCR, which means T cell receptors, when associated with MHC molecules. Okay, so they get recognized by the antibodies or T cell receptors. An antigen that induces an immune response, it stimulates the lymphocytes to produce antibody or to attack the antigen directly. So this all reaction due to this all reactions, even antigens are termed as immunogens. Okay, so don't get confused between antigen and immunogen, it is same, right? So these are two references which I had referred for this video. Okay, and you can go and check out these links. You may find some more information, okay, which can be. So next is, what are the properties or general characteristics of an antigen? The more foreign the antigen is to the body, the more immunogenic it is. So what does immunogenic means? It means it will trigger more immune response. Okay. Second is the chemical nature. So mostly antigens are proteins and polysaccharides. Okay. So why? Because they make a potent immunogen, which means strong immunogen. Okay. So a strong antigen which will trigger a good immunogenic response. Generally, the weight is 14 kDa to 600 kDa. Okay. So these antigens with these molecular weight, they are highly immunogenic, and one which are lower towards say 5 kDa of molecular weight, they are poor immunogenic. Okay, so generally that has come forward in some studies. Okay, so fourth point is susceptibility to processing and presentation. So if antigen cannot be ingested or processed and presented by APCs, it is considered as poor immunogenic, which means that particular antigen has failed to trigger a good immunological response. Okay, it simply means that. Next is dose of antigen. So there is a dose of antigen above or below which the immune response will be suboptimal. Okay, so if you have a good level of antigens getting invaded in your body, or they enter in your body accidentally or say by some other chance, okay, so depending on that dose, your immune response will depend. Okay, so if the dose is very low, you may not even, or the antigen will not even be capable of triggering an immune response. Okay, and if the level is good enough, then the body will show immune response. Okay, so in general there are two main divisions of antigens, as a foreign antigen, okay, and autoantigen. Okay, so the foreign antigens, they originate from the outside of body. Simple. So the example, it includes the parts of or substances provided by viruses or microorganisms such as bacteria or protozoa, as well as substances in snake venom, certain proteins in food, or components of serum or red blood cells from other individuals. Okay, So here I would like to give you a simple example that why we do blood group checking before doing blood donation. That is because even our red blood cells, they have A antigens, B antigens, correct? AB, and O has no antigens. Okay, so that's why we need to check before donating the blood. Okay, so that is the simple example. You can write that in your answers. Next is autoantigens. On the other hand, they originate within body. Okay, normally the body is able to distinguish self from non-self, but in persons with autoimmune disorders, normal bodily substances, they provoke an immune response leading to the generation of autoantibodies. Okay, uh, next is on the surface of antigens are regions called antigenic determinants. Okay, so it is similar to the epitope. Okay, so that actually feeds, or we can say it binds to the receptor molecules of complementary structure on the surface of lymphocytes. Okay, so that is the lock and key model between the antigen and antibody. Okay, so the part of antigen which actually feeds into a specific antibody. Okay, so the part of antigen is known as epitope, and the part which is present on antibody is known as paratope. Okay, so that actually works as lock and key model, right? The binding of lymphocytes receptors to the antigen surface molecule, it stimulates the lymphocytes to multiply and to initiate an immune response, including the production of antibody, the activation of cytotoxic cells, and or both against the antigen. Okay, so binding of antigen to the lymphocytes, it triggers the immune response. Okay, it initiates the immune response, right? So the amount of antibody formed in response to the stimulation depends on the kind and amount of antigen. Okay, so the same kind of immune response or the same amount of immune response will be generated to all kind of antigens. No, that is wrong. Okay, so the response or the immune response which is stimulated by each type of antigen is different. Okay, depending on the kind or depending on the type, depending on the amount or dose of the antigen. Okay, next are some terms which are related to antigens, and you may find these while studying your immunology course. Okay, so don't get confused between these terms. So first is antigenicity. So this is the ability of substance to bind specifically to the products of an immune response. Okay, so they will not induce immune response, but our blood contains some products of immune response. Okay, they are circulating throughout our body. So to those products of immune response, these antigens, they will just simply bind. Okay, they will not induce immune response, they will not trigger immune response reactions. Okay, so that is antigenicity, the ability of molecule to be recognized by antibodies or lymphocytes. Just the binding will happen. In case of immunogenicity, there is a little difference. That is the property of substance to induce detectable immune response. Okay, you can see here. Here actually the immune response is induced or it is triggered. Okay, the immune response can be humoral or cellular. Okay, so that depends. So actually when you see or when you uh, conclude that this particular antigen induces immune response, you term it as it has immunogenicity. Okay, the property. Next is tolerogenicity. So the antigen which is capable of inducing immunological tolerance is known as tolerogenicity. Okay, so immune system will not attack antigen. Your body will start tolerating that much amount of antigen. Okay, so what will happen? Your body defense system, that is your immune system, will not attack that particular antigen. Okay, next is allergenicity, okay, or simply allergy. Okay, so having these antigens, they have the capacity to induce allergy, okay, hypersensitivity. Next is antigen specificity. So this is the property of antigen or part its part to react with specific antibody. Okay, so a specific antigen will bind to a specific antibody only. So that is antigen specificity. Next is epitope or antigenic determinant. Very important term and generally used between antigen antibody reactions. Okay, so it is the part or portion of antigen that combines with the products of specific immune responses. Examples: antibody. Next is pro-antigens, so low molecular weight substances which do not induce formation but can cause delayed hypersensitivity reactions. Okay, so those are pro-antigens. The main difference is low molecular weight substances. Okay, low molecular weight antigens. Okay, so they will, what they will do, they will induce the formation of hypersensitivity reaction or a delayed hypersensitivity reaction. Okay, and last is heptane. So simply you can also write it as incomplete antigens. So a substance that is non-immunogenic but which can react with the product of specific immune responses. Okay, so heptanes are small molecules which could never induce immune response on themselves. Okay, so if only heptanes are introduced into a host body, then they will not trigger any immune response. But if they are administered with a carrier molecule, okay, then they are able to trigger or induce immune response. Okay, yeah, so here you can see there is epitope shown. So epitope is a complementary structure which is present on antigen, and it has complementary structure present on antibody. Okay, so it actually fits properly like a lock and key model. Okay, so some specific antigens, we will just have look out on this list. Okay, so general comments are like any molecule that is immunogenic must also be antigenic. Okay, but the reverse needed to be true in other cases. Okay, so it is like all enzymes are proteins, but all proteins are not enzymes, like that. Okay, so the first is proteins are the best antigens because of their molecular size and structural complexity. Second is polysaccharides like starch and glycogen are not good antigens because they are readily degraded within cells. Okay, next is lipids are poor antigens. Why? Because relative simplicity, structural instability, and rapid metabolism. Okay, so these are specific antigens which you should know. Why proteins are best antigens? Reason is given. Then why polysaccharides are not good antigens? That also the reason is given. And even for lipids which are poor antigens, why? Reason is given. Okay, so you can get these questions in MCQs. Okay, try to remember at least 1 or 2 points. Next is non-microbial antigens. Okay, so one of them is snake venom, then mosquito bite, pollen grains, serum proteins, then milk and food proteins. Okay, so milk and food proteins, these proteins will actually uh, cannot we cannot say that in each human being they will show uh, or they will trigger or they will induce immune response. Okay, because each individual is different. Okay, so it depends, and you may find that your body is also tolerant to some of these food or milk proteins, and you may find that your body is actually sensitive, okay, to these proteins. Okay, next is antibody molecules. Okay, so other antibody molecules, then histocompatibility antigens like in organ grafts, sorry. Next is bacterial antigens like capsule, K antigen or capsular antigen, cell wall antigen like O, somatic antigen, flagellum, that is H antigen, flagellar antigen, pili or pili, that is F antigen. Okay, so any of these antigens, if your body recognizes, the immune response will be triggered. Okay, then last are viral antigens. So capsid proteins are good antigens and they are highly capable of provoking antibodies. Okay, so we had covered our topic. Okay, so I guess yes, I had listed out all the basic points which are necessary to just uh, get your antigen topic revised before your exams. Okay, so thank you for watching. I hope this video is helpful to you all. So do like, share and subscribe to my channel, and do recommend your friends to even subscribe to my channel. Okay, thank you for supporting, and do follow me on Instagram and Facebook by the same handle, that is Microbial Concept. Till then, bye bye.